Hello there, I'm Chun Hong, the Chief Executive Officer of GISA. It's real privilege to make a presentation at Bio Europe 2022 meeting, and I'm happy to be here today to take you through GISA story. In this 15-minute presentation, I'm giving to you a brief look at our company, our cell products, and our corporate philosophy. So let me start by giving you overview of what our vision for future and what company is working on today to generate a safe, powerful, and affordable cell therapies with immune cell expander platform. Before we get started, let me tell you a little bit about our history. The company was started in 2018, shortly after our parental company GI Innovation was established. Since its inception, we have dedicated our efforts to developing immune cell products and to address current unmet needs in market. Headquartered in Seoul, Korea, we have offices, R&D centers, and cell and protein manufacturing facilities and capabilities. This gives us necessary tools and confidence to provide cell products throughout Asia and other countries. We offer both NK and T-cell products for treatment of solid and liquid cancer, and so far we have secured approximately 100 billion US dollars in financing. Our mission is to be a leading global company developing immune cell-based therapeutics and provide innovative solutions to challenges being imposed by existing therapeutics. And by doing so, we ultimately want to improve health and lives of patients and their families. The reason we need to do this is because we believe current immune cell therapies pose several challenges to be therapeutic for all patients. One of major challenges in this type of treatment is in cell manufacturing process. Because these are very complex processes and requires lots of work to generate enough cells, the product cost is very high. So with our technology, we want to extend the power of immune cell therapy by generating not only safe and potent, but also affordable therapeutic immune cells. With our technology, we also want to drive those therapeutic immune cells to target specific organs or deliver another therapeutic modalities to tumor cells. With all these desirable features, we believe GI cells products will address current unmet needs and contribute to maximizing therapeutic benefits for patients. As shown on the right, we are advancing our cell technology platform. Current focus and how we have developed the company, what we have raised funds for, and what we are pursuing every day is development of controlled and simplified manufacturing processes without genetic engineering or cancerous feeder cells through our immune cell expander platform. Our platform employs protein-based cell programming approach, and we will talk more about that as we go. In order to meet our goal that we have set up previously, we have assembled a world-class team of scientists, business people, and investors at GICell. I was the first one in after I spent many years working in the field of T-cell therapy. We have other team members who have extensive experience in immunology, cancer immunotherapy, protein engineering, and cell therapy together with manufacturing process development. They are the reason why we are pursuing many of these innovative concepts in cell therapy manufacturing. Moreover, our core management team has lots of pharma industry experience with proven track record in business and corporate development, and it's pleasure to have them on board with the leadership in GICell. Taken together, with all these combined capabilities and experience, we strongly believe GI cell can be readily positioned in the market and provide turnkey solution for the global community. But of course, none of this will be possible without investors, and we are privileged to have a number of very successful visionaries involved in our company. We brought in not only individuals, but also some institutions. Over on bottom, you can see key factors that have led to our success, and all these things are to build portfolio of safer, more potent, and highly scalable immune cell therapies. The list here includes obtaining core technologies for development of regulatory T cells and also nano-NK cells. In order to expand our pipelines, we have established many successful partnerships with global players to support our process development and also with research institutes to support studies for upcoming clinical development. Due to growing market for cell therapies, I believe GI cell is entering very sustainable market and is close to hitting inflection points as we anticipate filing of our first IND for allogenic NK cell this year. So far, we have raised over 100 million US dollars to focus on all our milestones and future activities. Immune Cell Expander platform is a unique product. It's unique because it's driven by ancillary protein-based cell programming. First, we generate novel combination of ancillary protein using our core protein engineering and expression technologies. Then we screen those ancillary proteins, giving best condition to immune cells for their growth and function. After this initial selection, we develop process to enrich immune cells and expand further in our manufacturing system. Our protein-based platform technology is also unique in that we have consistent read-through of biologic measures from in vitro through animal testing. Finally, consistent safety across clinical studies supports our platform technology. The first step in our platform is generating ancillary protein candidates, which are rationally convincing and structurally stable. In order to do that, we first select molecules from our libraries and in conjunction with GI cells deep learning technique, we construct series of ancillary proteins in specific format. These ancillary protein candidates are then expressed in our transient system and assessed for its property of matter and cultural feasibilities. Selected ancillary proteins are then incubated with PBMCs, our universal cell source for therapeutic immune cells. Following this 14 process, we harvest the cells and analyze them for high cell viability and consistent performance characteristics. Finally, we select ancillary proteins that are enriched with optimal features for our immune cells and further subject both protein and cells to scale-up process. Our manufacturing process is fast, consistent, and reproducible. Cells are sent to our manufacturing site for scale-up purification and expansion. Following this process, we cryopreserve final products, which create off-the-shelf NK cell or autologous Treg cell products, enabling treatment or retreatment for a longer period. The cells are those and now ready to inject into patients. Our entire process is with FDA-approved cutting-edge equipment and approach like Quality by Design is automated, closed, and transferable system that we have repeated many times. And we are very confident that this can be practical approach following successful commercialization and approval. As you can see, our platform is expanding. Our NK cell program is the most advanced, and we are planning to move this program to first-in-human clinical trials at the end of this year. And we are looking at advancing our cell therapy pipeline in regulatory T cells and other non-migrant diseases as well. By 2025, we believe that GI cell will be at clinical stage with several pipelines in the early and mid stage of clinical development. The characteristics of cells are very important. In addition to very high purity, cells are shown to express a high level of activation receptors and CD16, and this contributes to enhanced killing efficacy of our allogenic NK cells, called it TAP-NK. We screen donor samples to build pools of universal cell sources with optimal profiles which can be used for specific indication or treatment requirements for efficacy and safety. After this, we enrich and expand cells in our simplified process, which includes ancillary proteins instead of additional steps for gene editing or cancerous fetal cells. Furthermore, our data clearly shows that our platform can deliver very consistent NK cell products with prolonged expansion, which enables generation of high-dose and low-cost products. Again, data from multiple manufacturing runs clearly demonstrate that GI cell platform delivers cell products with consistent profiles. Here is a representative slide for manufacturing excellence of top NK cells, which present consistent production yield and marker expression profiles throughout multiple manufacturing runs. On average, we harvest around 100 billion cells, which yields around 100 doses, with 90% return from only one 50-liter batch. This clearly demonstrates that our platform consistently generates high number of cells from each run, thereby facilitating cost control for high dose as well as repeated dose NK cell therapies. Our in vitro studies against various cancer cells also support excellent potency of top NK cells. Our NK cells show strong tumor killing efficacy against leukemia, head and neck cancer, and colorectal cancer. In conjunction with top NK immunomodulatory profiles shown in previous slide, we believe our NK cell products have potential to be best in class, which will be demonstrated with further clinical development. Another program I want to introduce is drone Treg cells, which express organ-specific homing receptors on cells. And in this case, that organ is intestine. Due to enhanced expression of the specific homing receptor, our Treg cells are recruited in intestine and release high level of anti-inflammatory cytokines. This targeted action of drone Treg cells neutralizes harmful effects by immune cells. And those effects can lead to reduction of harmful inflammation and thereby increase repair of damages to intestine cells. We believe our drone Treg cells have potential to be therapeutic against the inflammatory bowel disease. I must say that we are fortunate to have great partners for clinical development in addition to supply partners. Now we have continued interest in working with companies where we can leverage our combined capabilities. Thank you for your attention. This is all I have for today.